It's my pleasure to introduce a new colleague here at ASU in the School of Life Sciences and also Biodesign, the Center for Immunotherapy, Vaccines, and Virotherapy, which is an acronym and center I'm just learning about, Dr. Esther Borges Florsheim. Dr. Florsheim got her bachelor's and then her PhD at the University of Sao Paulo in Brazil. After that, she went off to do a postdoc at Yale, where she won numerous awards during her postdoctoral studies. And she came here to join us just a couple months ago, actually in January of this year, to start as an assistant professor in the School of Life Sciences. Esther's work is, I think, a great fit for this seminar and our center, where she's trying to understand what has gone on in our immune system and why we develop allergies. She's an immunologist with core interests in how immunological defenses promote fitness and organism survival. She studies allergies, mast cell biology, and neuroimmune interactions, mostly in rodent models, to determine the impact of the immune system on physiology and our health. I haven't done this, I think, too many times before, but I like, when it's someone I know, I like to tell a fun fact about them. And since we've been talking a lot about New Jersey and the Northeast and people who love bagels in general, I just wanted to say an interesting fact about Esther is that she thinks bagels are weird and doesn't know why that we're maximizing, we're minimizing our bread intake because there are holes in them, right? Yeah. But I imagine that many of us would disagree with you, Esther, but that's just our upbringings. Yeah, see Kenny saying boo in the chat there. We have very, very strong bagel opinions in my lab and in general in the Northeast. But anyway, everyone, please join me in welcoming Dr. Florsheim, and we're very much looking forward to your talk.
Thank you. Let me share my screen and let me know if you can see or hear. And if something happens on the internet, just give me a shout because I cannot see the text. Yeah, I do think bagels are weird. And in my defense, I just saw a tweet the other day that was really good that's saying like, okay, enough argument about this. To eat New York bagels is just really good because you eat them in New York City, okay? So let's move on to my talk now. So thank you very much for the invitation to give a talk at this seminar series. I am a huge fan of the center and hope to start collaborating with some of you soon. As Noah said, I'm a new faculty at SOLS. I'm at the Biodesign CIVV. And as you can tell, I'm interested in the immune system and more specifically, the effects of immune processes in normal physiology. For example, the nervous system and behavior, which is what I'm gonna talk to you today. So I'll first discuss defenses in general and how it relates to the immune system, then talk about allergies, and I'll share the motivation underlying this research and explain the models we decided to use, and the experimental data that we got, the new ideas that we're proposing with this data, and finally, what I'm going to be doing here at ASU, my future projects. So in short, I'll show you that a noxious substance, for example, like food allergens, how can they change behavior through a neuroimmune mechanism? And don't worry, I know it's lunchtime, and for those of you who are having lunch right now, this is not particularly the behavior that I'll. So the conceptual framework of my research focuses on defenses, which are crucial for survival in suboptimal environments or unfavorable, negative environments. So environments can be unfavorable in many ways, as we know, by, for example, lacking essential resources, by not providing optimal social interactions, and by the presence of negative factors such as pathogens, predators, and toxins. So animals evolved a variety of specialized defenses to cope with these unfavorable environments, such as neuronal, behavioral, immunological defenses. And for example, pain, fight or flight responses, and inflammation are examples of these categories. So when I say immunological defenses, you probably think of antimicrobial responses. And this is a widespread notion about the function of the immune system that has historical reasons. And indeed, get any immunology textbook, even the most recent ones, and you will see that the definition of the immune system is related to protection against pathogens. You see the table of contents. That's what mostly immunologists have been focused on since it started as a field like many decades ago. So this is of course true. The immune responses are essential to limit the spread of pathogenic bacteria and promote survival. However, these encounters with pathogens are in general very rare compared, for example, with the encounters that we have most of the time. So the famous microbiome is, for example, harmless, and not only they are not dangerous at all, but actually most of the time beneficial to the host. So then the question is, if that's not what the immune system is doing all the time, then what is that the immune system is doing? Like, what are the roles of the immune system that are not antimicrobial? So we propose that whatever these new roles are, and I feel that this immunology field is going to have a boom of data in that regard in the next few years. What are the non-canonical functions of the immune system? And we propose that these new roles of the immune system should take into account the physiological effects that are induced during immunological events. And the second point here is that not only these physiological effects should be taken into account, but we think that they are also adaptive. They are also required for organismal fitness in response to this immunologically relevant stimuli. So immune defenses can in general be of two types. Type 1, which is what I just mentioned as like a antimicrobial, has antimicrobial activity, and type 2, it has antitoxin activity. And the strategy in type 1 immune responses or immune defenses is to eliminate pathogens. And whereas the type 2 responses, the idea there is to try to expose these noxious substances. We can make an analogy here. What's the difference between type 1 and type 2? So for example, if, well, let's say we were all in a normal seminar, we're all in one room and a mosquito comes in or a fly, this is the type 1 version of the analogy. And what is the best thing to do with the mosquito? You just slap it, right? Like that's the easiest way to get rid of that annoying annoying feeling. But let's say now it's not a mosquito that comes in that room, it's a skunk. A skunk comes in the room. You can try to kill a skunk, but it's probably going to be really messy and it's probably not going to go very well. So the best strategy in that case is to try to get that skunk out of there without too much noise and too much like chaos. So that's the exact analogy for type 1 and type 2. In type 1, you want to eliminate the pathogen directly or the cells that are infected with the pathogen. And in type 2, there's nothing to kill directly. You want to try to expose that noxious stimuli. Important point here is that exacerbated defenses can lead to disease. And I'm going to go back to that point many times in my talk. So for example, exacerbated type 1 defenses can induce sepsis, for example, or autoimmune diseases. And we think that exacerbated defenses of the type 2 category can lead, for example, to allergies. We decided to focus on food allergies more specifically to study the immunological defenses, and mainly because the GI tract is a major mucosal site for interactions with the environment, and it's also a major immunological site. Almost up to 80% of the immune cells of your whole body reside in the small intestine alone. So we decided to work on, our model is in the gut. So what are food allergies, right? Like we probably all heard of that. It's becoming increasingly common now, especially in kids. So we know exactly what is the effector part of the response, like how does the immune system contribute to these reactions? What are the symptoms that allergic people have? Like you have increased vascular permeability, bronchoconstriction, GI motility, and mucus production. The symptoms can vary, can be like a very light, like skin rash to lethal anaphylaxis, for example. It's highly prevalent, and the problem is that it's increasing in modern urbanized Western societies. So just here in the US, and this is actually old data from 2011, is that like around 10% of the kids in the US right now have some type of food allergy. So what do we know about it is that those are exacerbated reactions of the immune system to food antigens. And food antigens are mainly in this 8 categories here. All these foods have major allergens. And the main question is why we have allergies. That's what bothered me since finishing undergrad, that I've been working with allergies, is because of that. Like, how can we have evolved a whole system to just make us feel sick? Like, we don't know what's the role of, for example, mast cells or these immune cells that are participating in allergic reactions that are not to just induce allergies and make us feel sick. But that's like a not very satisfactory explanation for that. So I think the explanation for that, and I really like this idea, again, coming back from what is the difference between defense and disease. So when we say allergies, most of the time, we are referring to this left part of this curve. It's the allergic diseases, which is like, it's an exacerbated chronic response that induces pathology, right? Like that's the disease. So within this category, food allergies are obviously there, asthma, atopic dermatitis, drug hypersensitivities, and so forth. But the idea is that there are some physiological counterparts of this exacerbated reactions, right? Like we didn't evolve this to induce allergies. They are a consequence of exacerbated defenses. And these defenses are mostly unknown, and that's exactly what this project is about and my intended research. And what are the allergic defenses? Like, what does our body evolve to do to protect us from noxious stimuli that is actually protecting us, is not leading to disease, right? So we can cite some that could potentially be involved in that, some neuronal reflexes, some detoxification pathways, some activation of like immune cells, like eosinophils, mast cells, and so forth. So this leads us to the main questions of this project. Number one is what types of defenses are induced in this type 2 immunity, or more specifically in allergies? How does the immune sensing of allergens can affect physiology? And the third one, I'm not gonna answer, and that's for my future projects, is are there any fitness consequences of triggering excessive defenses? And what about no defense at all? So that's for our future, we can discuss at the end. Interestingly, when you think about allergies, you realize by just thinking of the symptoms of that people with allergies have, is that they usually overlap with neuronal reflexes. So for example, itch, vomit, sneeze, and avoidance behavior, for example, aversion, right? Those are symptoms that people with allergies often display. And we thought that that was a good start for us to go through, like, to decide which allergic defense we're going to study. And I particularly like the avoidance behavior the most, the food aversion the most, because it's even more strategic in terms of you don't even need to get into contact with the noxious substance. It's even smarter that way to try to avoid future exposures and not even have to get sick or anything. So we decided to look in food aversion more specifically. So the first question is, can allergies even induce food aversion? And these are the aims of this project. So to characterize aversion in food allergy, to determine the immune sensing mechanism and how the gut inflammation contributes to aversion, and identify brain responses to allergens. So this is the model. I'll just spend some time here for you to understand, like, how do we even induce allergy in mice? That's our current model. Different strains of mice can get allergic to egg white protein, or called ovalbumin, or ova. It's how I'm going to refer here. So basically, you sensitize mice. You just give this protein combined with an adjuvant on day 0 and day 7 through the subcutaneous route. There are different ways of doing it. We compared with other routes as well and other adjuvants, but for this talk, I'm just gonna show you this OVA antigen with an adjuvant. And then to test food aversion later on when these mice are allergic sensitized, we're gonna test their avoidance behavior by a very simple preference test. So just a little bit of like basic immunology, like what's happening when you're giving the adjuvant, right? Like why are we triggering this allergic sensitization with this adjuvant? It's mostly unknown, like long story short, we don't exactly know how this adjuvant works in this case to induce type 2 immune response, but we know that certain adjuvants, for example, aluminum hydroxide in this case has been used for that. And what happens is that you induce the production of antibodies called IgE. These antibodies are produced by B cells in lymph nodes in wherever you give them. And then you just produce this allergy type of antibodies called IgE. And this IgE, it's in the circulation, but they will eventually sit on mast cells. Mast cells are tissue resident cells, and they are all over your body, and they're just normally living there. And they always have some type of IgE sitting on them. But the point here is that this IgE is specific to the protein that you associated with the adjuvant. So this IgE, for example, would recognize ovalbumin if you give that later. And the other point here is that this mouse is not allergic per se, it's just sensitized. Like it's potentially allergic if they get in contact with the allergen in the future. So this is our first set of experiments. I'm gonna summarize a lot of the data, but this is important to go through a little bit more in detail. So this is a very simple assay. So mice are already sensitized. There are control and allergic sensitized. And 2 weeks after the first sensitization, we test for what they prefer to drink, basically. So where it says day one there, the mouse in the cage, they're individually caged. It's a special cage that we built. It has like an electronic surface with like metal stands underneath each bottle. So every time that they lick one or the other bottle, we can measure that in number of licks. And you can imagine how much bullying I suffered during my postdoc with this project, but that's okay because the science still prevails. So the thing is that in control mice, and you will see what happens, like, so they all have a choice. They can drink from the water bottle or they can drink from the ova bottle. The ova is nothing more than just a protein shake. It's just egg white protein in water, right? It's a protein shake, literally. So we do this for 1 day. The next day we wash out the bottles and give just water in the two sides. And the third day we reverse the size just to make sure there's no place preference. And it's indeed like avoidance or preference. So we just reverse the sides of the bottles. And this is what happens with control mice, like right in the middle. Normal control mice, if you give them the choice of drinking from a protein shake versus water, they obviously, as you can tell in the orange curve, oh, I realize I have my mouse here. Here, this curve, can you see my mouse?
Yep.
Okay. The orange curve here shows the preference, how many licks. This is cumulative licks over time, and these are done overnight. When they're mostly active and hungry. But control mice prefer to drink from the ova bottle, obviously, because it's way more interesting, more caloric, more, it's a protein shake. So they have never seen that before, it's great. But if you had previously sensitized mice with that strategy that I just showed you with ova plus that adjuvant before that, then they change their preference. You can see here on day one already that they have a slightly different behavior towards that ova bottle. They will start drinking, but then they start drinking water a lot to try to dilute that. And this is on day one. What happens on day three is that this gets even more exacerbated, and this is this configurates like a true like avoidance behavior. The first one you can say, oh, it's a lack of preference, and which also can be defined as aversion. So allergic sensitization does induce avoidance behavior or food aversion in mice. And this graphs on the right side is just showing the first 60 minutes, because I was interested in knowing how quick this happens. Like if you, like this system that we have measures licks every minute. So we can have a very good sensing of time of how long does it take for them to start avoiding. And it seems that it's immediate. It's like within the first 20 minutes, if I even go narrow down to the 20 minutes, they already know that there's something there, which is really interesting. Like if you think about again, I sensitized these mice in the subcutaneous, like under the skin, like in the back of the mouse. That's how I sensitize mice. So this is the very first time that these mice are being exposed to the solution, and they already have this different type of behavior. So this is fascinating and super fast, relatively fast, and it's already a robust response. Here I'm gonna just like summarize, I'm not gonna show you all this data, so just summarizing all that we have about this avoidance behavior or food aversion in mice is that it's dose dependent. We know the concentrations that induce that. There is like a hypersensitivity feature to that because, for example, if we start with the dose like 0.1% of ova, that does not induce avoidance. It only starts inducing avoidance after 0.3. But if we go 0.1, 0.3, 1, and then a week later, put the 0.1, again, they will now avoid. So you create like you increase the set point, the threshold that you now can recognize the presence of that allergen, which is very interesting. There is memory. We sensitize mice and then we start doing avoidance behavior every other month. And as far as we went, which was 50 weeks after sensitization, which if you think about is like half of the mouse's life, they still had avoidance behavior, like getting even more pronounced every time. So it doesn't go away. Even if you don't challenge, you don't do anything with those mice, they have this memory of aversion for a long time. It's very robust. Like if you try to trick them and give sugar, for example, in the ova bottle, because mice absolutely love sugar. And if you add sugar and create now a conflict of if they drink, it's something that they really like, but there is the allergen there, like what happens? They will still not drink the allergen, which is very interesting that this food aversion is very robust. That means that this allergen is a very negative signal, perhaps. And also if we deprive them of water and they really need to drink, they will still not drink as much as the control if there is the allergen in that solution. This avoidance is specific to that allergen. If we use a different type of albumin called bovine serum albumin or BSA, then they don't avoid at all. They will all the groups will drink a lot. So it's very specific to that particular protein. We think it's adaptive because if you force mice to only drink ova, like let's say there's no water, I will leave ova bottle for like more than a week now. What happens is that they will decrease the solution intake by half compared to controls, and they will lose a lot of weight. So they really don't want to drink that. They will prefer to be sick than to drink that thing. And it's very broad apparently because we thought that that was a type 2 immune defense or immune response, but it's also applicable to a different type of adjuvant. When we give, for example, LPS, or like it's a bacterial molecule that induces type 1 immune response, this also promotes avoidance to ova, which is actually very interesting. It's more universal than we think. It's not specific to type 2 responses. And the anecdotal story here is that it seems that this is true for humans as well. And this is the only time that pediatricians or doctors come to me after my talk and say like, oh, we see exactly that. That's the first time that they agree with a basic scientist that works with mice. And they say this always, like they see this in clinic very often. Sometimes the kids already know that they're allergic and they start avoiding the food before the mom even realizes, or the dad, that they have any type of like food allergy. And the other story is that when they're undergoing immunotherapy to try to get rid of that allergic inflammation, 50% of the kids will not even be able to do immunotherapy, which would be effective. And it's because they avoid, like the idea of immunotherapy is to give crescent doses of the allergen, but like very tiny to increasing amounts over time. But kids avoid it, so they don't want to drink that, they don't want to eat, they spit it out. So you cannot even do immunotherapy very efficiently because of this food aversion. So it's a thing as well in humans, it seems like. So the second aim now is to look into the immune response and what are the immune mechanisms and how does the gut look like. So as I told you, what happens when you sensitize mice with this type of adjuvants is that you induce a type 2 sensitization and you induce these antibodies called IgE that will now bind to their high affinity receptor called Fc epsilon R1 in pink in the figure, and in cells such as mast cells and basophils. So the first thing we try to do is to ablate the cells and see what happens with the avoidance behavior. And the way to ablate, we got a model that all these cells that express, there are either mast cells or basophils that express this receptor for IgE. We can deplete those cells now in a diphtheria toxin dependent way. And we did that after they were sensitized to not mess with the sensitization. And here's what happened. So those mice are called RMB, red mast cells and basophils, because they're also reporter. But in any case, the wild type mice, again, as we saw before, they like to drink ova. And if they were previously sensitized with ovanalum, now they reverse. They start liking, but then they start avoiding after a while. If you deplete now the cells with diphtheria toxin for 3-4 days, what happens is that the controls continue liking ova, which is important. That means the mast cells and basophils are not important for this preference to protein, but their preference changes to, their aversion changes to ova. And now they do not like, now they reverse their aversion and they start behaving as like control mice and preferring to drink ovalbumin compared to water. And we kept doing this type of approach. Like we now used mice that were deficient in IgE, for example. And the same thing again, just a summary, mice that are allergic, as I showed you before, have decreased preference to ova. If you deplete, if you have mice that are deficient in IgE, this is now back to the same as the control. So they start liking ova again. And the same is true if you ablate or if you have mice that are deficient in the high affinity receptor for IgE, this Fc epsilon R1. This is in a different strain, which is good because also shows that in a different strain, there's also avoidance behavior when they are allergic, but the deficient mouse reverses this avoidance and behaves as a wild type. So this brings us to this idea of like defense. That's where I think we are manipulating this. That's where I think this model is about, is at this stage, the mice are showing like a protective behavior, right? Like to be able to avoid a noxious stimuli, it's most likely adaptive and important. And at this phase, the only things that we know that we have so far is just the production of this type 2 antibodies, mostly IgE. There's this other allergy related antibody that I haven't mentioned, but it's also there. And there is the avoidance behavior, but nothing else so far. If you now try to look at the disease state, which is the next one, right? Like, are the same mechanisms going to be at play to induce the disease? Because that's what we're looking for, like same mechanisms that are the physiological counterparts for like a disease state. So how do we do the disease state in this case? It's pretty simple. Like mice are already allergic and sensitized, the same with humans. If you get if you're sensitive to bees and you got stung, the first time that you get stung, nothing happens, but you have to really watch out for the later ones because you now build up this allergens and you're going to be hypersensitive and potentially anaphylaxis. So what we have to do now is just to expose mice on purpose to the allergen through the oral route by gavages. So we just give like a lot of, this is like a full established protocol to induce food allergy in mice. And now you can see a lot of gut inflammation. And this is what's happening now. You're inducing the migration of inflammatory cells to the gut, and that's how. And now your mast cells degranulate. There's the sensing of allergen through IgE. There's the granulation. There's release of a lot of mediators in the bloodstream, in the nearby area, including the famous histamine that is one of the responsibles for anaphylaxis. So here are the readouts that we can, I'm not going to show you any of this data, but just to like, this is all the things that characterize this allergic inflammation. So presence of type 2 antibodies, way more now, you increase by a lot by challenging mice with the allergen. Increased transit time and gut motility, there's diarrhea, accumulation of mast cells, they move and they go to the gut or they proliferate. There is eosinophil migration, which is another readout for allergic inflammation, potentially anaphylaxis, increased mucus production in the gut. You have cellular infiltration and proliferation in the lymph nodes, in the draining lymph nodes, type 2 cytokines, which are immune molecules that are characteristic in this type of inflammatory responses. And the length of the gut changes. Okay, can you hear me?
Yep.
Okay. In any case, the point of this slide is to show that there are a bunch of readouts that are present at this stage, but not all of them are induced by the same things that induce the avoidance behavior. And that's why I put the IgE knockouts. So IgE is important for the ones in bold. So for anaphylaxis, for mast cell accumulation, for diarrhea and increased transit time. But mast cells and IgE are not important, for example, for all the other readouts. So we can summarize this here as like at the disease state, you have all these readouts, but those are the only ones that are IgE dependent, for example. And that earlier, if you look at the defense part of this process, IgE is important for a protective behavior, but if it gets continuously exacerbated and chronic and in a situation that the mouse cannot choose what to do anymore, then the same type of mechanism is now going to be responsible for the pathological, for some of the pathological effects that we see that are involved in allergies. The last mechanism that we found, that we just recently found to be involved, is super interesting, and it's related to this molecule called GDF-15. So the idea to look at the GDF-15, so this is a growth factor from the TGF-beta superfamily. And it's a very interesting molecule because it's just recently being found a lot of roles for it. And it's one of the markers for all kinds of diseases. Anything that you can think of had been published to acutely induce GDF-15 in the circulation. Anything, you can name any disease you want, GDF-15 is going to be up. And our group, which is the left, that's the graphic abstract from our group, they showed that in type 1 inflammation, GDF-15 was induced and was important for the physiological adaptations in that particular model. So we decided to also test in ours. And other groups have shown that GDF-15 is inducing nausea, which is super interesting. And that's why we thought that that could be a candidate. And indeed, GDF-15 is increased just 1 hour, probably less. We're trying to look at even shorter times now, but it's definitely induced by allergen challenge. And now if we give an antibody after mice are sensitized on day 14, for example, if you give a shot of antibody to block a GDF-15. You do this right before doing the behavioral test, you see that the behavior now changes. So all these mice are allergic sensitized, but one received isotype control antibodies, the other ones received the blocking antibodies. The one that did not, the isotype control behave as expected, they avoided the solution of OVA, but now the mice that got the GDF-15 blocking antibody start liking OVA again, and this is pretty quick change. So we are guessing that GDF-15 is a key mediator of avoidance behavior in our model. And the last set of data that I want to show you is the brain responses. What's happening if there is a change in behavior? There's presumably the information has to be being sent to this central nervous system, right? So that's what we did. Like, how does the question was, how does the brain look like after allergen exposure? So the same thing, we sensitize mice day 0 and day 7 with OVA and alum, and we challenge these mice. And right after we challenge these mice, we collect the brains and look at neuronal activation by classic CFOS. And then we can analyze all these areas that are activated in allergic versus controls. This, I'm showing some of these areas here. And they, interestingly or not, they are exactly the types of areas that we were expecting based on other types of data with other noxious stimuli that causes aversive behavior. So those are nucleus of Tractus solitarius, parabrachial nucleus, lateral hypothalamus, and central amygdala. There are some other areas that we're confirming now. Area postrema for sure. So this is like a summary of all the data that I showed, so we can discuss a little bit, and I'll show you what I'm planning to do now. So in short, the allergic sensitization induces a robust, very stable and long-lasting food aversion. This aversion depends on this GDF-15 molecule, mast cells, and IgE, and it activates central pathways that I just showed you, and it's depicted here in the figure on the left. So how does this information go from the gut to the brain? That's the next mystery to be addressed. Like here, we propose different ways like how this could happen. The top one is that some circulatory factor induced by mast cells is inducing avoidance behavior. It's going to the brain directly and acting on areas that are outside of the blood-brain barrier, and that can induce avoidance behavior. That would be the simplest. And GDF-15 experiments point to actually that mechanism because it's a circulating factor. And the only receptor that we know of that bind GDF-15 is in the area postrema in the brainstem, which is exactly an area of the brain that is outside of the blood-brain barrier. So that's our current hypothesis. The second one that we're still trying to address, but this one is the most technically challenging, is to see if sensory fibers are involved in this transmitting this information. So we're trying to target like vagal afferents or dorsal root ganglia sensory fibers. And the third model is that it would be maybe a combination, or maybe there is like an intermediate, maybe it's not the mast cell factors directly that will induce this avoidance behavior, but instead there is like an intermediate step here with a different organ. And we came up with this idea because GDF-15 is mainly thought to be produced in the liver and in the kidney. So for now, that potentially is that's our hypothesis for now. That could be that some mast cell factor triggers something in the hepatocytes, for example, that secretes GDF-15, and that goes to the brain somehow. They're not mutually exclusive. Obviously, you can have all of those things happening at the same time, and I wouldn't be surprised if that's the case. So if the sensory fibers are anyhow involved, then we have an interesting situation here of like a new modality of sensing the environment, right? Like the way we right now think we sense the environment is through our senses, our classical 5-6 senses, right? So basically through our nervous system. That's what these things in gray represent. So the first is olfaction, the second is visual system. The third is auditory system. So the way they work is that there is direct sensing or sensing through a modified epithelial cell of signals in the environment. If it's smell, like something, the light change or the air changing, the vibration of the air changing, but they're all sensed by sensory fibers, and they send this information to the central nervous system to trigger appropriate behaviors. And what we found is pretty much the same type of, it's analogous to the situation, because you do have environmental sensing through an immune factor, or through an immune system here, which is the antibodies, IgE in this case for allergen, that sits on mast cells and recognize some chemical noxious substance. And then potentially there is the transmission of this information through a sensory fiber that goes to the brain, to these areas that we saw, and triggers avoidance behavior. Now this neuro-immune module is interesting, and it's different than these other ones because it's not hardwired as the first three or all the other senses that we know of are. Like, you are not born with that. It's not innate. You have to, you create the specificity to what you want to be sensitive, do you want to respond to upon demand. So, and the other ability is that the immune system is able to create like tens of thousands of different types of specificities to everything you can think of. So you can think that this system amplifies this sensing module by a lot. So that's an interesting point. The last part of this discussion is just to talk about like more on food and diet selection by the immune system. These are ideas that we recently published in this perspective article, if you want to check more in depth. But the main point is that we obviously know that there are food quality control systems, right? Like if you have a nutrient versus a toxin that you eat those things, there is some systems in the gut, in the GI tract, a lot throughout the GI tract that determines if you should consume that, or if you should digest that, or if you should avoid or expose that. Our main idea here is that the immune system functions as such food quality control system, and all these immune defenses that I showed you fit into this category of food quality control. So it decides if things are bad or not, and obviously the organismal state influences this a lot. If animals are hungry, if animals are sick, if the environment is such that you can't eat or not, this all influences this decision. And we're going to argue in this perspective that those food quality control systems are the ones that can get exacerbated and derailed in food allergies, for example. And it seems like a trivial problem to select for food, but it's not. Like, it is trivial now that we can go to a grocery store and I can pretty much eat whatever I want if I go to that grocery store, because everything was pre-selected. Everything that is there is probably of good quality. It's enhanced flavor. It was made to make me feel attracted to that, right? But now if you think of a normal, natural environment, which is the one in the left, like if I'm thrown inside of this, in the middle of this jungle, what am I gonna do? What am I gonna eat? I don't know. Like I didn't grow up in this environment, and like, how do you decide in that environment what you eat? So the main point here is that the immune system is playing a key role in determining and helping you to make that decision. So again, coming back from to this defense and disease states, that's exactly what I'm interested in. I'm going to keep pursuing this type of line of investigation to find out what are the defense mechanisms that are the physiological counterpart of diseases. And as Noah said, I just started at ASU. I'm at the biodesign building. And my major interest is exactly to discover this new non-canonical functions of the immune system that are not antimicrobial responses, but something else. Like, what is the something else that the immune system is doing like all the time, most likely? And I think it mostly affects like other physiological systems. So for example, as I showed you, it could be affecting behavior and the central nervous system. So we would like to explore different types of behaviors here, or what are the other defenses that are triggered by immunological stimuli. And others, like we could I can cite like metabolic changes or hormonal changes that are physiologically relevant to be adapting to this new environment full of immune stimuli, right? And this is the short-term goals, is to obviously define what are these mast cell derived mediators that can induce this type of behaviors. If there are the sensory pathways, to identify what are the specific sensory pathways inducing, linking the immunological sensing to the behavioral response, and to go into more of the central nervous system and define what are the circuits that are responsible for this immunologically mediated behavior. And as I mentioned, one of the most interesting parts here is to explore a little bit more of the behavioral side of as a defense, as a physiological defense. So we all have experienced in our lifetime when we get sick with some bug, like a virus, a bacteria infection, how we feel about that, right? Like we trigger this type of reactions that induce anorexia. So you don't want to eat, you just want to sleep. You have like this transient depression and you don't want to exercise at all. So those are all modifications, changes in behavior that are actually protective. Like you should do those things in order to get better recover from infection faster. They're probably all adaptive. And the last one is obviously, this is for type 1. We know a little bit more about, because we have always like felt something like this before, and it has more of that on the literature. And the last one is type 2 immunity. What happens when you trigger this other type of immune response, which is antitoxin, which are probably more present in the environment than eventual pathogens. So what happens in this case? And that's my last slide. Going back from like, what are these non-canonical responses, and that we should consider this physiological response. So we know, as I showed you, excessive defenses can induce inflammatory conditions that are detrimental. For example, sepsis, autoimmune diseases, allergies, that all comes from this side of the response, which is exacerbated immunological responses, so exacerbated inflammation. Now, if I'm telling you that immunological stimuli can also induce different physiological responses, what are going to be the costs of inducing this type of physiological defenses? Like if this gets chronic and you are in an environment that has this toxic substance, this irritant all the time, and you cannot get away from it, what's going to happen with this? So one idea is, for example, to explore the behavioral aspect of that. And that was greatly inspired by Professor Randolph Nassy from the center, by his last book, Good Reasons for Bad Feelings. So is it possible that mental illness have any immunological basis to it? That means, can we try to predict and do it in an experimental way, in an empirical way, to see the development of some mental illness, for example, anxiety, obsessive compulsive disorder, depression, whatever it might be? We can try to guess depending on the immunological stimuli that was induced. So that's what I have for you today. And I hope we can discuss a little bit about that. I just want to give a shout out to my course that I'm teaching this fall. It's all related to exactly what I told you. Is the immunological origin of diseases, in which we're going to explore all these like non-canonical functions of the immune system. So tell your undergrads and grad students to join me next fall. Check it out, the course description there. And that's it. I would just thank all the groups that I was part of that made it possible to even get to that point and get all this research done. So I was a postdoctoral researcher at this lab, the immunology lab the Madzitov lab, and I collaborated a lot with this neuroscientist group from the Dietrich lab, and that's it. Just contact me if you have anything. Thank you.
Thanks, Esther. That was fantastic. So I'll open it up for questions.
If the internet allows.
Right, if you're on campus, and the internet allows you to ask questions, you can ask. I have lots of questions, but if anyone has, who's not going to stick around for the class later, if you have a question, raise your hand, or Stephanie, go for it.
Hi, Esther. Nice to see you. Really enjoyed your talk, and this is such cool work. I'm so excited to see all the things you do. It's probably a bit far in the future, but I was wondering if you have plans, or if the team that you were working with has plans to assess the cortical areas for like higher order sensory processing in connection with the development of the avoidance behaviors, because I noticed that the amygdala was, CFOS was lit up in the amygdala, and that's the key region for developing like fear conditioning and aversion. So just kind of wanted to hear if that was in the plans for the future.
It's in the plans for sure, but I don't have expertise in higher order. So that will depend on collaborators at ASU. That's a really good, interesting question. Like, how does that change decision-making, for example? Because for now we're talking about like the old areas of the brain, right? Like, it's something that probably very primitive or ancient types of systems can do. It's just like C. elegans definitely does have avoidance behavior in a way. Like if it's in contact with pathogens or some noxious things, they just retract. So you don't need a higher order brain to be able to do that. But that's not to say that there would not be any implications in those, in the systems that have this higher order brain. So that would be interesting. I was thinking of looking at the insular cortex, because that's where most of like immunological reactions would affect. Perhaps, but that's definitely something for a collaboration type of project.
Awesome. Well, we have great behavioral neuroscientists over in psychology across the street. So
let's do it.
Sure, Federico Sanabria would be especially interested, and others. So yeah, thank you. Look forward to working with you.
Thank you.
Other questions? I'm going to ask mine. So you have these two different mechanisms, GDF, like molecular mechanisms, GDF-15 and you said IgE on the mast cells, right? And they both could be working, GDF-15 is expressed in the hepatic cells. Have you tried, so you did an anti-GDF-15 and you also did an anti-IgE, right? Have you seen if the effects are, it wasn't anti-IgE, it was
anti-IgE is complicated. It doesn't work as well, and it's interesting that that's true in clinic as well, because you give anti-IgE and it's something about the antibody, and like it can actually activate a little bit. So it's not a good approach, but we are doing the knockouts for GDF-15 to compare with this. So we have both of them. It's still preliminary, repeating the second experiment for the GDF-15 in deficient mice, but it was the same phenotype. So
was it any different? Because like if was it any different? So like if the mast cells are upstream of or signal, are affecting the signaling of the hepatic cells, the GDF-15, GDF-15, like doing both, treatments to reverse this aversion behavior, was it stronger with the two of them, or faster, or longer lasting?
Oh, I see what you're saying. Yeah, if you give them together, right? We have not tried that, and maybe because the IgE antibody is horrible, but we can do the anti-GDF-15 in IgE knockouts. So to see if it potentiates the avoidance behavior, right? Yeah.
Yeah.
I have a feeling that this GDF-15 is not the only thing that it's, and the timing is funny. I didn't show you that much of the kinetics, but GDF-15 doesn't prevent avoidance right away. It's delayed. So I think it's not the main thing yet. I think there is like a neuronal pathway. We did have some preliminary data with the vagus nerve, which is what I'm betting but it's really tricky to really find out if it's a vagal afferent pathway. We did a subdiaphragmatic vagotomy, but like it has so many criticism problems in doing this approach that it's like a technical thing. But I feel that there are the neuronal thing that sends the information right away and starts inducing this avoidance behavior. And I think this GDF-15 comes later and is responsible for more sustained avoidance. I don't know.
Got it. That was going to be my other question, is how quick this, like what we think is like the rapid signal for this aversion. And so you think it has to do with the vagal afferent nerve?
Yeah.
And
Yeah.
what, I mean, what's stimulating the nerve and why? Like, and how?
That is a good question. Yeah, that is a good question. And I don't think it's GDF-15. We are trying to look if mast cells produce a GDF-15, and it doesn't seem like it, at least in all the experiments that we've done. We did RNA-seq, we did, anyhow, it doesn't seem it comes directly from mast cells. And that's why I put that third model that there is like an intermediate tissue there to do that. I'm highly betting on the liver right now, but just because of precedent literature showing that that's where mainly the GDF-15 comes from. But what from mast cells, I can tell you a lot of the things that do not induce that, because we tried like all the common things that you can think of that mast cells have, like histamine, serotonin, leukotriene, prostaglandin, all kinds of things, substance P, CGRP, all the neurotransmitters that we were able to try to pharmacologically target, we did, and it was none of those things. So it's way more tricky. That's where we're going to try to find out eventually. That's right.
Cool. Thank you. Any other last questions before we give Esther a quick break? All right, well, everyone, please join me in thanking Esther for a wonderful talk.
Thank you.